Hey, hello everyone. Welcome to the eighth installment of our high elevation 5 needle white pines 2024-2025 Science and Management webinar series. I'm going to be your host today. I'm Karina Lee, a master's student in Diana Tombach's Forest Ecology Lab at the University of Colorado, Denver. And I am co-organizing this webinar with Lou Deloisi, a PhD student in Daniel Ulrich's lab in Montana State University, and Enzo Martelli, a PhD student in Carr Nelson's lab at the University of Montana, who also happens to be one of our two presenters today. But before we get started, I'd like to introduce board member Kara Nelson for a few announcements for us. Welcome, Kara.
Thanks, Karina. The webinar today marks the end of the third year of the Whitebark Pine Ecosystem Foundation's webinar series. And the goal of the series, as you all know, is to share knowledge about both science and management of all high elevation pines, including whitebark pine, but also to create a forum for networking. So towards that end, feel free to introduce yourself in the chat, mention any work you're doing, anything you want to do for networking. Another goal of the series is to promote the work of the Whitebark Pine Ecosystem Foundation. If you are not a member, please consider joining so you can support the foundation's work. And a link will be posted in the chat. Each month before the presentation, a board member gives a brief update on one of the foundation's initiatives. So today I'm giving that update, and my job today is simply to close out the webinar series, which means first and foremost, thanking our early career organizers, Karina, Enzo, and Lou. I want to thank each of you for your efforts to deliver a high quality webinar every month. I also want to thank our speakers for their excellent presentations. This year we had Amanda De La Torre, Michael Murray, Kristen Waring, Lacey Hankin, Randy Moody, Kira Hefty, Sharon Hood, and then today Enzo Martelli and Vlad Kovalenko. So really appreciate your participation in the series. I have a special gift I will be mailing to each of you in the next few days. If any of you missed any of these presentations, all of the videos are up on our YouTube channel. There'll be a link posted in the chat. And if you subscribe to the channel and hit the notification button, then you will get an alert every time we post a new video. So if you're not able to attend next year. So my last piece of business is just to announce the amazing news that our 3 early career organizers are staying on with the webinar series for next year. So I'm gonna really thank all of you and give you a floating clap here in appreciation that you'll be sticking around for next year as well. So we'll be back after the summer break. Registrants will be receiving a survey for inputs and feedback to help organize the series for next year. So that will come maybe in a month or so over the summer, and then we'll have a calendar for next year out in August. So thank you to all the participants for creating this community, and again to our organizers and all the speakers.
Awesome. Thanks for that, Kara. Personally, it's been my pleasure. This has been really fun. So after the presentation, we will have a Q&A session with our speakers. This is a really great opportunity for discussion, so please don't hesitate to put your comments and questions in the chat box. And so now I'm really excited to introduce today's speakers, Enzo Martelli and Vlad Kovalenko, because not only do I work with Enzo on this webinar series, but I've also worked with Vlad back when he was a master's student at the University of Montana, and I was a biological technician in Glacier National Park. So I actually first heard the story of the whitebark pine and the Clark's nutcracker mutualism at a talk that Vlad gave 4 years ago for the Glacier to Medicine Alliance. And he did such a great job that I decided to study Clark's nutcrackers for my master's thesis. So, and then that summer, I also helped Vlad with his fieldwork by going out to one of his remote sites and checking on an autonomous recording unit, which you're going to hear a lot about in this presentation. And I know for a fact that the results from his master's work are already being used by government agencies to inform nutcracker monitoring, which is a really great achievement. So Vlad is currently a biologist with the U.S. Fish and Wildlife Service and a research associate with the Nelson Restoration Ecology Lab, which leads me right into my introduction of our very own Enzo Martelli. Enzo's research in the Nelson Lab focuses on applied science with particular emphasis on the effects of forest management and restoration practices in high elevation ecosystems. As demonstrated by his collaboration with Vlad, Enzo is a team player through and through and has worked with a variety of partners from land managers in the Forest Service to undergraduates to the Society of Ecological Restoration, and then obviously this series and WPEF. So I can say from my experience working with Enzo that he is constantly thinking of creative ways to bring more people into the discussion surrounding conservation of five-needle pines. And he's really been an inspiration to me this past year. So with that double intro, I'm thrilled to welcome Enzo and Vlad to our final installment of this 2024-2025 webinar series to talk about the effects of restoration on the primary seed disperser of whitebark pine on the Sawtooth National Forest. Welcome.
Thanks so much for your introduction, Karina. Can you see my slides?
Yep, they look good.
Yeah, cool. All right, so good afternoon. Thanks to all for coming today. We're really happy with the participation. We got this fourth year of the webinar series, and as Kara and Karina mentioned, we wanted to close it with a collaboration project where Vlad and I work together along with an incredible team from the Nelson Restoration Ecology Lab at the University of Montana, the Forest Service ranger stations in the Sawtooth National Forest, and the Institute for Bird Population. So during the next 40 to 45 minutes, Vlad and I will present you preliminary results of the research projects on whitebark pine restoration monitoring. And it's an interesting project because we use an autonomous recording unit to identify Clark's nutcracker habitat use, and this is going to be used for integrating into understanding better how these treatments affect ecosystem recovery and response. So the project is titled Understanding the Effect of Restoration on the Primary Seed Dispersers of Whitebark Pine on the Sawtooth National Forest, although our presentation today will be focused primarily on what have we learned from the use of ARUs, or autonomous recording units. So to start with this, something really important is that over the last 30 years, in the field of ecological restoration, we're experiencing an increase in the number and also the extension of projects and research. And along with this trend, there is also an increase in need for reliable and generalizable information on the efficacy and effects of these management practices. Even more, when this information is being used to inform massive restoration initiatives like the United Nations Decade on Ecosystem Restoration, or the 20 by 20 initiative, among others you might be familiarized with already. And for this reason, it's really critical that the restoration projects that are aiming to inform about the effects of the treatments do implement designs that are capable to detect effect and separate this effect from other factors related to environmental variability, site-to-site variability, year-to-year variability, and all those combinations. So for forest restoration, it is particularly important that we understand how changing environmental conditions and specifically ecological disturbances can affect ecosystem response to these management practices. And it's even more important trying to understand how stochastic events such as wildfire or mountain beetle outbreaks or diseases can impact the treatment effects and the capacity of the design that is being implemented for monitoring to detect a response. So like pretty much how much is going to be watered down by that spike. And in this context is where whitebark pine and its recent listings makes a perfect candidate to try new innovative strategies that can help us expand what we already know in terms of ecosystem response to management interventions. And over the years, we've all heard it, multiple strategies have been developed across the species range for specific habitats or specific needs. But among all of them, one of the most influential has been the range-wide restoration strategy for whitebark pine that was developed by Bob King and his collaborators about 13 years ago. Yet, this specific strategy has been I would say the one that has seeded all of the ones that follow in terms of what conservation or management strategies to use, and that are even included in the National Whitebark Pine Restoration. And why is this important? Well, it is important because this strategy calls primarily two main types of restorative practices. On one hand, we have mechanical removal of shade-tolerant competing species, and on the other hand, the use of prescribed burning to emulate wildfire regimes on whitebark pine communities. But all these practices try to aim in one way or another overlapping objectives relative to recovering spatial heterogeneity, promote natural regeneration and diverse age class structure, and also in general terms, improve stand health and maintain ecosystem functioning. However, outside of a few forest types, and this is common by now, there is relatively little understanding of the ecological effects of these practices for restorative purposes. And this information is specifically lacking for forest type without a commercial value, pretty much like whitebark pine or high elevation ecosystem that experience less frequent mixed severity and stand replacement fires. On the other hand, the ones that we do know a lot about are located in dry, low to mid elevation coniferous forests where thinning and burning treatments have been broadly implemented, mostly for reducing fire hazard and increased stand resistance to wildfire. So for this ecosystem, there is not much of a problem. There is a large body of literature on treatment efficacy and effects, but this also leaves us with a huge opportunity to further develop our understanding on the effects of these practices in whitebark pine communities, but not exclusively to whitebark pine, pretty much for every species present on subalpine conifers. Subalpine conifer forest. Yet, developing effective monitoring programs to understand these treatment effects is challenging in general, but especially so for species that exhibit high spatial variability and complex regeneration dynamics, which when we start thinking about whitebark pine, both of them apply to it. So being able to reasonably make inference on the effects of the treatment, it is critical to assess the efficacy of the monitoring design and to adapt the design when necessary and as much as necessary. So given how important maximizing efficiency is for managers and practitioners, because they oftentimes need to balance generating precise information, but also working with limited funds and personnel. So in this context, we're aiming for a long-term approach that can separate this. It's really important. And the best one that we know to date is the design named before-after control impact. That pretty much should be the golden standard, specifically because this design is capable to tell us the magnitude of the effect through time, even more depending how often do we come back and resample those very same units. Anyway, we've all been there. Sometimes even using the most robust design and having all the resources can fail when we're trying to understand whether these practices are effective or not. And it's not necessarily the fault of the design. Sometimes it's just because at the time with the information we had, we didn't aim directly at how to answer the questions we had on mind, whether we selected the right variable, whether we had the right level of replication, or whatnot. And in systems as complex as whitebark pine communities, we do know that that is a constant. So to illustrate this, I'll share with you some interesting results from a 15-year after treatment assessment that we did on the ecological effects of restoration treatments on whitebark pine. And for this assessment, we used Bob Keen's long-term monitoring project, Restoring Whitebark Pine Ecosystem, which was the first project to use a replicated bucket design. And it was directed to test the effects of selective thinning and prescribed burning as restoration treatment. This project was established when I was barely a toddler in the mid-90s, and it included a combination of experimental mechanical cuttings, pre-camp burnings with or without mechanical cuttings, including control units as well. And this original study was implemented at 5 sites across the northern Rocky Mountains of the United States, specifically western Montana, in the Bitterroots National Forest, Salmon National Forest, and Clearwater National Forest. But for this specific example, I'm only going to be focusing on Bear Overlook. something that I need to mention here is that the majority of these sites at the time of the establishment of the plots were in their later stages of forest development, with varying levels of mountain pine beetle mortality and blister rust infection. And this is something that we will remember in a couple of minutes, or that we take into account in a couple of minutes, relative to what we found. So among the multiple response variables that we study, we were interested to see the effect of treatment on adult tree mortality. In many ways, trying to answer the question of like, are we killing more trees than we should be killing? And for that, I followed the trajectory of all the live adult trees that were tagged during the establishment year, and that was like in 1993 or something like that, and followed their development until 2015. Within the control units and all the treated units. The treatments that were implemented in this experimental unit were prescribed burning and prescribed burning with a combination of mechanical cutting. And the way that you see that in the panels are, in the x-axis, you'll see C that represents the control unit, Rx that represent the prescribed burning unit, and Rx plus M that represent the prescribed burning unit combined with mechanical cutting. On the right side panel, you'll see the difference of whitebark pine light mature trees in an index, in an individual per hectare metric. And on the left side panel, you will see the whitebark pine mortality percentage change from the first year to the 15th year. And what we found is that neither of the treatment had a significant effect on adult tree mortality, and that this could be the result of many factors. As I mentioned earlier in the introduction, might not necessarily be because the treatment didn't have an effect. There is way more to tell there that could teach us like what to do best next time. So, but being some of those factors, the most probable ones, related to having maybe a too harsh initial condition for this type of restoration strategy. So for this type of active restoration strategy, when we have a high incidence of mountain pine beetle mortality plus a high incidence of blister rust infection, might not be the case. Although with the data that we have today, we can't tell that specifically. And the other case might be that just for this specific type of response variable, which is adult tree mortality, that, as I mentioned also earlier, has a high spatial variability, maybe the monitoring design was not the adequate for this specific process in the way that the data was collected, or maybe the replication level. And there will be an article coming out in the following months that delves deeper into that aspect. Similarly, we were interested to see the effect of the treatment on natural regeneration as seedling density. And what we found is that after 15 years since treatment, neither of our units showed a mean increase in seedling density. And in the contrary, like the regeneration still seemed to struggle to get to pre-treatment levels. It even seems that it like decreased. And this could also be the result of many factors, but most of all, the ones that we believe could be answering that is that stand conditions after treatment were too harsh for nutcrackers to come and be seen back again the units. And if we relate this to the previous result, the amount of adult tree mortality was so high that maybe by this point there was barely 1 or 2 trees that were producing cones. If so, and without cones, or with a really low density of cones, the ones that were not subjected to pre-dispersal predation by like squirrels might not have been like enough to see a strong regeneration pulse after the treatment, even 15 years, 30 years or so after the treatment. So in order to overcome some of the limitations we had with this project, the Nelson Restoration Ecology Lab, led by Kara, decided to start a new long-term monitoring project that would be also the starting point of a restoration network that was introduced last year during the Baker City Conference that can inform about the ecological effects of these treatments, but other combinations of treatments such as planting and whatnot across the range of the species. So for this project, as I mentioned in the beginning, we collaborated with the Sawtooth National Forest and the Institute for Bird Population to create a new protocol that would expand on the lessons we learned from Bob's project. And for this, during 2023 spring, we evaluated prior monitoring data in order to develop a new protocol. And during that summer, we went to the field and we collected some data to analyze whether this new monitoring design was going to be strong enough to provide sound information about the questions that we're going to pose. And last year, we redesigned the protocol using that data from pretreatment data, or using pretreatment data. And after making the assess of the efficacy of the protocol, we came back and established the permanent plots with this new strategy. So for this project, we selected 3 conditions representing the most common stand structures in the region, pretty much central Idaho between Stanley all the way through Fairfield in the south. And each condition would have 2 experimental units, one a control and another one on a treatment, where each experimental unit would be composed by 10 plots per unit. These plots were located using, as you can see in your screen now, a grid to accommodate for complete stand sizes and shapes. And in this part of the region, that is the rule. It's really difficult to find a single area that extends with a constant cover of whitebark pine dominating the canopy. And some of the variables that we collected were trying to focus to answer questions on the density of seedlings and sapling by species, growth rates, density of cones, frequency of insect and disease, fuel loading, cover of target and storage species, which, as you can tell, is pretty much a comprehensive approach, and collecting that data would take a really long time. But what makes this project different or more innovative is introduction of a new monitoring technique, which is using ARUs to estimate habitat use for Clark nutcracker. And with this, I'll leave you with the expert on this, which is Vlad, which will explain the importance of this method, these methods, and the findings that we got. So with that, I'll leave you with Vlad.
Thanks, Enzo. And thank you so much to Karina for that lovely introduction. I didn't realize that I was the one to introduce you to this ecosystem, so I'm very honored and also very excited to see your results of all your work. So I'll share my screen, and would someone please jump on and let me know that you can see my slides?
You look good.
Thanks. All right, so yeah, as both Karina and Enzo mentioned, my name is Vlad Kovalenko, and I was privileged enough to be part of this pilot season last summer in the Sawtooth National Forest. Acoustic recording unit, the human and autonomous avian surveys compare. So a little bit of background on acoustic monitoring. It's an emergent field in wildlife studies, and so far it's been used in several different ways. It's been used to infer site occurrence and occupancy of cryptic and rare species, such as the spotted owl. It's been used to assess biodiversity and ecosystem health. It's been used to look at the occurrence of marine mammals. And I used it in my master's work in Glacier National Park, looking at nutcracker habitat use of whitebark pine stands. And so from that work, I don't know if you can see this figure well, I also kind of compared in-person point count surveys, which is the figure on the left, with vocalizations recorded by autonomous recording units, or ARUs, which is the figure that's on the right. And as you can see from these data, when you don't have a lot of nutcrackers occurring at a site, you have very wide confidence intervals when you model their occupancy at those stands, because there's not a lot of certainty as to whether or not the birds are truly using those areas. And so we were hoping to kind of remedy that by getting a much larger sample size using acoustic monitors, which unlike people who can only be out for a few days a summer surveying birds, those acoustic monitors could be out for months at a time, collecting data continuously. And so when we compare these two data sets, we can see that in relative terms, the confidence intervals around the expected number of vocalizations at sites is quite a bit tighter, but some sites still do have quite a bit of variability, as you could tell there at the top. And we'll get into the various reasons for that here later in the presentation. And so for my study in Glacier, the biggest takeaway was being able to relate whitebark pine stand characteristics to nutcracker habitat use. And unsurprisingly, we found that cone density had a very strong positive effect on nutcracker occurrence in those stands. And so the objectives for this study and pilot season in the Sawtooths was to first assess the appropriateness of our design with ARUs. We wanted to be sure that we were adequately placing the ARUs distance-wise from each other so that we could achieve sampling independence. We wanted to make sure that we were sampling the correct window, and we were particularly interested in the whitebark pine cone harvest season, which is typically in late summer. However, there's some variability in that as well, and we'll get into that a little bit also. Also, we wanted to assess the reliabilities of our ARUs and our ability to use them to actually be able to estimate the mean number of nutcracker vocalizations at the stand level. And then last, we were curious to see how our acoustic recorders did in comparison to observers on the ground counting birds. And so getting into the methods for the point count surveys, the protocol we followed was to do a 10-minute survey at each of our ARU sites where we documented all the birds that we saw and heard, as well as their distance from us. And we did our best not to double count any individuals. So we did 3 surveys per site, and those surveys coincided with ARU deployment in the beginning of the season. Then in the middle of the season, it coincided with ARU maintenance, where we swapped out memory cards and batteries. And then the final survey of the season took place while we were also removing and retrieving the ARUs from the field. And the data from these surveys can be used to inform occupancy models, as well as density and abundance models. Moving on to the methods for the acoustic monitoring, we deployed the Cornell Lab of Ornithology's Swift and Swift 1 recording units, which is this little white box strapped to a tree. And we had 5 locations per experimental unit. So among the 6 experimental units, we had 30 recorders total. We tried to space the recorders at least 100 meters apart, as I mentioned, in order to maintain sampling independence and not double count birds. These recorders were out recording from 5 a.m. to 9 p.m. every day, starting in early August and ending in early September of last summer. After we got all the data back from the field, we ran the raw recordings through a machine learning algorithm called BirdNet Analyzer, which identifies all the vocalizations in the recordings to species. And then, since for our purposes here, we were only interested in nutcrackers, we filtered out all the other classifications. And then these data can be used also to inform an occupancy model, as well as vocal abundance and density models with some limitations, which I will discuss. So jumping into the results from point counts, most of our point count surveys did not result in nutcracker detections. And as you can see, there amongst the 6 experimental units, there were not very many detections at all. This last unit, salt bounds treatment, the rightmost bar there, had no nutcrackers detected in point count surveys throughout the entire season. Those are some summary statistics for the point counts. And here's a little histogram. So as you can see, mostly non-detections, and the most nutcrackers ever detected in a single point count was 2. And here's a histogram showing kind of a breakdown of the time of day that our point count surveys took place. And I just wanted to show this to highlight some potential sources of bias in our point count data. So the standard point count protocol procedure dictates that you should have your point count surveys done within 5 hours of sunrise. So that's kind of probably right around 11 to 11:30 right there. And as you can see from this histogram, we mostly did okay with that, but due to some logistical difficulties and some technical difficulties with recording units, some of our surveys did end up being well outside that window. And so that's just something to keep in mind with any sampling design or project. Some unexpected things can happen, and suddenly you're not really surveying at an ideal time. So moving on to the results of our acoustic monitoring, nutcrackers were detected by acoustic recorders at all sites and units. And here you can see kind of a distribution by experimental unit of the nutcracker detections. And as you can see, there's quite a bit of spread in the data. And the sites varied quite a bit, especially these 2 lower vocalization sites on either end. Those are some summary statistics. As you can see, our mean was a lot higher than our median, so we had quite a bit of skew in the data, and we had a very high variability. The good news, we had very high classifier precision of 0.97 out of 1. And classifier precision is the number of true positive vocalizations divided by the sum of the true positives and false positives. So a false positive would be if a wind gust is classified by BirdNet to be a nutcracker call. And our collaborator, Mary Clapp, who's on this call, actually painstakingly validated these data by listening to 300 random Clark's nutcracker classifications and manually annotating them to make sure whether or not they were true nutcracker calls. And so 292 times out of 300, our classifier was correct. So that's pretty good. So now we'll assess some of our study design metrics. So starting out with ARU placement. We found that ARU spacing was mostly adequate, and we did a pretty good job at spacing them out to be 100 meters apart or greater. However, due to some of the terrain heterogeneity and impassability, some of those ARUs did end up being a little bit closer than 100 meters, which is not great, but it is what it is. And so Mary found that on 13 occasions, birds were double counted by 2 recording units. So a bird made a vocalization and 2 units picked that same vocalization up simultaneously and counted it. And then on 3 occasions, 3 recorders picked up the same vocalization. So that's a little concerning. However, if you think about the fact that we had a total of almost 20,000 nutcracker detections, 16 double counts is not too bad. Next, we assessed the timing of our sampling design. So here on the left, we have the total counts from point count surveys. at all the experimental units, broken down into the first, second, and third survey period. So early, mid, and late season. And now on the right, we see the same data from acoustic detections. And from both of these, we can kind of see that we start out high, and then as our sampling period progressed, the detections decreased at each unit. And so these results suggest that our sampling started too late. We would want to see kind of a gradual increase, then a peak, and then the decrease that we see here, not just a peak and a decrease. And I kind of had a feeling when I was out there in the field deploying these recorders that we might be of the harvest season there. So next, assessing our ARU's reliability and ability to estimate mean vocalizations at the stand level. So our vocalization data, as I mentioned before, is highly skewed and variable. And so that makes it pretty difficult to estimate the true mean. And Karen Nelson did a power analysis for this sampling methodology and found that we actually had pretty low power at all of our units. And so in this figure, you have the frequency of vocalizations on the x-axis and the relative margin of error on the y-axis. And as you can see, even at our most precise or best sampled site, we still had a margin of error over 70%. And so that kind of tells us that if we're trying to
Keep losing Vlad here.
Yeah, I was just gonna say, Vlad, if you can hear me and you come back on, maybe we could have you repeat that because that was an important sentence.
And I can fill in too while we're waiting for Vlad to come back, that what we found was that our ability to estimate the mean number of calls at the stand level was not so good overall. Vlad, I was just re-explaining this graph because you blinked out for a minute.
Can you hear me? Can you
Yeah, we can hear you now.
Okay, sorry about that. I guess my internet isn't very good. I'll stop sharing video just to help that. All right, back to this. Can someone tell me what the
So we heard you for this reliability, but now the slide's not showing.
Yeah, you were just saying that the best sample site had a margin of error of over.
last thing they heard me say was?
Oh, there's a little bit of lag
Okay.
now. Can you hear me, Vlad?
Can you hear me?
Yep.
Yes.
Okay.
Yeah, so the last thing I heard you say was that the best sampled site had a margin of error over 70%.
Right.
And then we lost.
While you were blinked out, I did a little bit more interpretation.
Okay, great, thank you. Continuing on, I guess I was gonna say that I'm not overly surprised by these findings because the bird that we're studying here, Clark's nutcracker, is especially during the cone harvest season, it's neither sessile nor does it have consistent vocal rates. And so, for example, here we have a savanna sparrow, and if we were doing acoustic monitoring of savanna sparrows during their breeding season, we would expect to have much higher consistency in our data because savanna sparrows will basically just find their spot to nest in a grassland, and the male will stay at or near that nest for the entirety of the season, and sing with relative consistency, and you'll be able to have a more predictable vocal rate and lack of large-scale movements by the animal. That is not what we have with Clark's nutcrackers, and that could be a limiting factor of using ARUs to study them. So next we compared biologists versus ARUs to see how the two methods compare. So for point counts, we only had 25 out of our 90 surveys that actually yielded nutcracker detections. And as you can see with the total detections by site, we missed nutcrackers at quite a few. 13 sites out of the 30 did not have any nutcracker detections by humans. On the other hand, acoustic monitoring, the recorders picked up nutcrackers at all sites, and so did quite a bit better as far as consistently picking up birds, which is unsurprising as they're in the field for a month at a time, unlike us. And so we wanted to explore a little bit the causes for these mismatches in detection. And so we found that during point count surveys that did result in nutcracker detections, only one ARU picked up birds at the same time and place. And on the other hand, during point count surveys where the observer did not detect nutcrackers, there was one occasion where an ARU did. And so here's a little figure kind of breaking down the presumed causes for this mismatch. We concluded that most of these mismatches could be attributed to factors such as distance from the ARU, where 15 point count detections were greater than 80 meters from the observer and the recording unit. 3 of those detections lacked vocalization by nutcrackers, and so they couldn't be picked up by the unit and were only seen by the observer. And then 3 more detections were during high wind, and so there was likely a lot of acoustic interference on the microphone, and so it couldn't hear those bird calls. Then 4 more observations by point counts. We couldn't explain why the ARU didn't pick them up. There's probably some other variables that we didn't account for that caused that. So now I'll just discuss the takeaways from this study, from this pilot season. First big one is that we should start sooner because it seems pretty clear from our data that we missed the start of the cone harvest season, and we may have even missed the peak. Next, we found that point count surveys and acoustic monitoring can be complementary to each other. Point count surveys are obviously going to pick up birds that don't vocalize or for other reasons can't be heard by the acoustic monitors. And on the other hand, acoustics are complementary to point counts because there's much more consistency and simply much more data collected over the course of sampling season. Again, because you simply can't have an observer out for a whole month at a point. And then, of course, we found that nutcracker stand level metrics can be really difficult to estimate using ARUs because of their high mobility and the high variability in vocal rates. And here's a diagram from another paper. Amanda Naveen and others were looking at these rare birds in Hawaii, and they were actually able to use the ARU recorded call density to predict the density of the birds in their habitat. And as many people who are in wildlife know, density is kind of the gold standard of understanding animal populations. And this figure just kind of highlights the relationship between how frequently these birds vocalize and how many of them are actually at a site. And that is not the relationship between nutcracker vocalizations and their occurrence, unfortunately, especially during the cone harvest season. Oh, and then lastly, we learned that our classifier, BirdNet Analyzer, is very good at identifying those nutcracker calls in recordings. And so that's very encouraging from the standpoint of not having to manually verify and validate those recordings and just being able to trust the classifier. So next steps going forward, we're going to play with these data and try some other modeling approaches, including the one from the paper in Hawaii that I just mentioned. It would be cool to compare our point count density estimates to kind of relate them to call density if we can. Here is a figure of some modeled call densities at our 6 experimental units. So definitely excited to explore such avenues of data analysis. We could also relate our calls to habitat covariates from whitebark pine characteristics that Enzo and his crew collected last summer. And here's one where I just compared the cones to the predicted vocalizations. And as you can see, there's definitely a relationship there. Surprise. And then, with the richness of the data that acoustic monitors collect, it'd be cool to look at some other interesting metrics such as species richness. And here's just a small snippet of some of the different species that were detected by our acoustic recorders at our sites. And then, of course, we'll try to publish our findings. So be on the lookout for that paper. And then finally, I would love to just acknowledge some folks who made this work possible. Kara Nelson and the Nelson Restoration Ecology Lab for having me join this project. The crew that was out there who are really hardworking and definitely know how to have fun. Mary Clapp from the Institute of Bird Populations, who really did the bulk of this acoustic analysis. The Whitebark Pike Ecosystem Foundation for contributing funds and having us on this webinar. Of course, the Sawtooth National Forest for collaborating with us and letting us do our sampling there, and the U.S. Fish and Wildlife Service and Forest Health Protection for funding large portions of this project. And with that, I am happy to take any questions. Thank you.
Thank you so much for your talk, Enzo and Vlad. I would like to now open up this opportunity for our attendants to share any questions and comments you might have. We have about 12 minutes left, so please go ahead and type your questions and comments into the chat. And if we don't have time to go over all of your questions, don't worry. Someone from our team will send them over to Enzo and Vlad with your contact information so that you can further discuss any questions that you might have. While I wait for folks to think of questions and to pop them into the chat, we do have a question from Karina. It's a pretty specific question, so I'm going to go ahead and mute myself and then let her ask it.
Yeah, so this is right up my alley, seeing as my project involves point counts. So one of our transect data is all collected by going out there and doing this exact protocol that you were describing, Vlad. So really great to hear more about it. I guess one question I had for you is, how did you manage to get distances to nutcracker vocalizations? Because this is something that we kind of shied away from doing just because it sounded difficult. Yeah, I was just curious, like, what your thoughts are on that.
Yeah, it is really difficult, Karina. And great question. It was a total estimate. A lot of the times you can kind of ground truth yourself because you've got a rangefinder in hand. And so if you hear a bird vocalizing and then you're able to see it like in a similar period of time, then you can get a pretty accurate estimate. But yeah, the times that you only hear the bird and don't get to see it, you're basically just relying on your brain's ability to process the volume and assign an estimate for the distance.
Yeah, that's kind of what I imagine. And I also imagine that, you know, the terrain might play a role as well in terms of your ability to hear a bird. And then they have so many different vocalizations. So it's like, yeah, you might be, you know, accustomed to the volume of their like crack call, but then what happens when they're doing their like bullfrog call or something like
Yeah,
that?
no,
So yeah,
totally.
definitely tricky.
Yep.
And then I'll just kind of keep rolling. I think it's so great that you did this work. I think it's something that many of us have wondered about, like whether this was possible. And one thing I was hoping you could do for our audience, Vlad, if you don't mind, is to just give like a really brief explanation of what occupancy is, because I think it's important that our audience doesn't walk around away from this thinking that ARUs are not useful for monitoring Clark's nutcrackers, because you know I think they totally are. And then I guess as sort of a follow-up to that, you mentioned that there might be some really good ways to integrate like point count method and ARUs together. And I'm wondering if you, or Mary, it's good to see you, Mary, by the way, if you have any thoughts of ways to do that. Because this is such a great tool, especially when you consider like, you know, these are remote sites that we just can't be at all the time. And there's got to be a way that we can use this technology to our advantage. But yeah, that difference between density as sort of like the gold standard and occupancy, I guess, would be useful.
Yeah, so I guess I'll start out talking about occupancy and density. And Mary, please jump in here and add anything I missed. But occupancy basically is if you have a site and you're trying to determine whether an animal occupies it, or I guess in our case with nutcrackers in the cone harvest season, occupancy is a bit of a generous term for what we can assume they're doing there because they are so mobile and they rarely spend too much time in one place. So for this, I would say I would downgrade occupancy to occurrence. Do these animals occur at this site? And so if you're using point count surveys exclusively and you just happen to not be there at a time when the bird is, you might conclude that that site is not occupied or that the target species doesn't occur there. And then on the other hand, when you have such a huge sample size as you get from acoustic data, yeah, and you run an occupancy model on, like if you were to run an occupancy model on this data set, you would conclude that 100% of our sites are occupied or that the animal occurs at 100% of these places. And so the desire then becomes to try to get something of a more meaningful metric out of those data. And that's when you start wanting to do something like density and actually be able to count the number of individuals based on your acoustic data. And as I mentioned, you know, for some species, that's actually being done. Will it happen for nutcrackers? I don't know. Probably not in the cone harvest season. Maybe during the breeding season, but nutcrackers breed early, and so you'll have to be skiing into those places and you know, maintaining your acoustic recorders during rough weather and all that kind of stuff. So lots to think about moving forward. And yeah, go ahead, Mary.
I'll hop in and just say that this is sort of one of the challenges, but also one of the great opportunities of using ARUs is that on the one hand, the data streams that result from all of this, like more or less continuous monitoring, don't neatly fit into the current statistical methods we have to determine something like occupancy or abundance, as Vlad so neatly described. On the other hand, if you pair acoustic monitoring with in-person visits, whether they're point counts or nest searching efforts, getting to John's question maybe, or grid searches, not only do you have this sort of permanent acoustic record of what has happened there, but you have in-person observations that you can then use to correlate those behavioral observations to different types of patterns of vocal activity. So not just is the species there and is it vocalizing, but is it doing a particular call that you only hear at nest sites or on their breeding territories, which we're developing the sort of machine learning methods to do, to sort of do those subspecies level identifications. But without the in-person corollary data, we're sort of left guessing what these different patterns of vocal activity actually mean biologically. And so I think that is a really important part of using acoustic monitoring going forward. Yeah, an exciting avenue, especially for nutcrackers, because the question, maybe it's not quite so easy to answer the question of occupancy, but with a little bit of effort, we can maybe answer a more meaningful question, which is like, how many, what is the sort of metric of stand use for Clark's nutcrackers at a given stand, and what are the stand characteristics of those whitebarks? And that's actually, you don't need to assume occupancy in order to answer that question. You just need to use the right data and the right statistical model. So anyway, I'll stop there.
Thank you both, Vlad and Mary, for that really detailed answer to Karina's questions. We do have some audience questions and a couple more minutes, so I'm going to go ahead and ask you one of those. So from Odin Scholes, they wanted to know if it was possible to use the ARU detection data to come up with a length of time that a point count would need to ensure nutcracker detection. And you touched on this a little bit, but just to make sure that we're getting at all of the audience questions.
Yeah, I think that's a really good question. And I think the answer is yes. And I will defer to Mary, who is much more statistically savvy, if you wouldn't mind fielding it, Mary.
Yeah, I think if you were to create a sort of accumulation curve where you sort of like count the number of Clark's nutcracker vocalizations that you get every minute, every consecutive minute of recording until you start detecting them at any given site, you might find that you would go hours or days before you detect a vocalization, whereas at other sites, you might detect one in the first hour of your recording. And so I'm not sure that data would be able to directly inform, that data might suggest that you have to do 4-hour point counts, which wouldn't necessarily be feasible. But on the other hand, they could inform you about the sort of like hourly probability of vocalization, but you may, as we've discussed, those rates would vary. They do vary markedly by time of day, by season, by stand level characteristics, and so you need a lot of like site level replicates in order to estimate that sort of hourly probability of vocalization. But you could do that.
I just wanted to jump on and say also that having the ARUs to figure out when to do the point counts is extremely useful. The number of minutes of point counts you need would depend on if you need to estimate to the stand or just to that point. And then a final point about that stand level estimation, which was even difficult for the ARUs. However, at the peak frequency of calling, the ARU estimation was pretty good relative to the outlier time periods. But there's a lot to think about there in terms of how to make the point counts function and the ARUs function.
Great. Thank you all so much for your detailed answer to that question. And thank you to the audience for asking such great questions. We are at 1 p.m. Mountain Time. So on behalf of the Whitebark Pine Ecosystem Foundation and the webinar organization team, I want to thank Enzo and Vlad again for your participation. Our next webinar series will begin in the fall, and we look forward to having a whole new set of presenters. And thanks again, everyone, for attending, and stay tuned for our upcoming installments. We'll send that out closer to the fall, and see you all next year.
Thanks,
Thanks,
everyone.
everyone.
Bye, everyone. See you next year.
Bye. Have a good field season.